Hello, I'm Jose Saavedra, Medical and Scientific Director for Nestle Nutrition. In this section, we'll discuss gut microbiota and immunity, and most importantly, its implications on pediatric health. We'll first describe what an intestinal microbiota is. I'll describe how the intestinal microbiota support a healthy immune system, and understand as best as we can the factors that affect bacterial colonization of the intestinal tract and their implications on immune system development. The intestinal microbiota is a very complex ecosystem. It's composed of hundreds of species. We estimate somewhere between 500 and 700 species populate the GI tract. And they exist in very large numbers, roughly 10 to the 10th. That means 10 zeros of colony-forming units per milliliter of intestinal contents, particularly in the distal GI tract. Very importantly also, the GI tract is home to most of the cells of the immune system, what we call gut-associated lymphoid tissue, or GALT. It is this relationship between bacteria and the large number of immune cells that is important for an infant to adequately develop its defensive mechanisms or barrier function and adequately modulate immune response. It is the bacteria in the GI tract which are critical in the development and maturation of our immune response and gut barrier function. Let's briefly review a little bit of these mechanisms. Bacteria function in many ways in terms of their relationship with the intestinal mucosa. In terms of supporting gut barrier function, for example, it is bacteria which stimulate the production of mucus and mucin, a very important part of this first layer of defense of the intestine from the bacteria that populate the GI tract, particularly those that we call pathogens. The GI tract also decreases its permeability when bacteria are present, meaning less large molecules can go through. And this increase in the tight junctions between enterocytes, which decrease this passage, is in response to the presence of bacteria. Bacteria in the lumen also support the development and maturation of GALT. For example, bacteria will stimulate GALT to produce secretory IgA. And very importantly, bacteria also help balance the responses of T helper cells, both T helper 1 and T helper 2, as well as T regulatory cells, which are critical for immune modulation. The intestinal microbiota, or this microflora in the lumen of the gut, evolves over time. Children are born with a gut that has essentially no bacteria. It's practically sterile. And it's just over the first 72 hours of life that these bacteria dramatically increase in numbers in the intestine of an infant. These first 72 hours are critical in determining the profile of bacteria that will compose this child's intestinal microbiota. There are a number of bacteria and ways that we could define or divide these microorganisms in terms of the types and species that grow. We have a number of facultative anaerobes and anaerobic bacteria. And very importantly, there's one particular type of bacteria we call bifidobacteria. It's a specific genus that tends to predominate in the gut of a number of infants, particularly those who are breastfed. Over the next few weeks, this intestinal flora becomes more or less stable, and the number of species becomes stable, with slight changes later when a child weans, for example, and starts eating other types of food. But it is extremely important to understand how this change in the intestinal content actually makes a difference in terms of immune system.